So we continue Unit 2 with a discussion of membranes. Now, we're going to start by reiterating some things about phospholipids. Phospholipids, as you'll recall, are going to have a phosphate group and a choline group as part of our hydrophilic head, with the glycerol here as the backbone, and our fatty acid tails will be over here. 2 fatty acid tails and a phosphate group and a phosphocholine head. So hydrophilic head, hydrophobic tails. Hydrophilic head, hydrophobic tails. Now, if you followed along the live version, this is what's going to happen here. At the water's surface, when you drop some phospholipids, which I'm going to draw as this, if you drop those into a water's surface, right there, I just dump some in. What's going to happen? The water is going to interact with the surface, and the tails are going to be sticking up out of the surface. So the heads will interact with each other, and the tails will interact with each other in the different surface. The non-polar air and the polar water. Now, if we're underwater, if we are underwater, under the sea, as it were, and we have nothing but phospholipids, then we will have our phospholipid heads interact with each other to protect each other from the terrible, terrible perils of the outside world, forming a micelle. This micelle will have the hydrophilic heads facing outward and the hydrophobic tails facing inward. This cannot be particularly large. We can increase the size of a micelle by first adding more, but eventually that will reach a point where it won't matter anymore, where you'll just break it up and have a bunch of small micelles again. So what we do instead is we can form a bilayer of infinite length. It can go on forever and ever and ever and ever and ever in both directions, but that would take infinite phospholipids. And last I checked, that would take infinite resources. And one of the problems with living in this world is that we don't have infinite resources. So I believe that is not a possible thing to do. So what do we have to do to make this bigger? We need to make it into a circle, a lipid bilayer circle. So, I've already drawn out of ink. So, with a hydrophilic outer layer, hydrophilic inner layer, and a hydrophobic tail inside inner membrane space, within the membrane space. So I can't draw too well in 3D, but 3D is how this happens. A bilayer sheet will be a continuous sheet of these phospholipids going on forever and ever in all 3, up, down, side, side. But really this is just in an XY plane anyway. We've got micelles, which range in size from 2 to 20 nanometers. And then we have liposomes. Liposomes, fat bodies in Greek, which are a bilayer where you have removed some of the lower layers so that it kind of curves in on itself. Now, of course, this has been split in half. This continues as a sphere with another smaller sphere inside. Same with this side, sphere. So that's a liposome. This is not yet a cell membrane. A membrane requires a couple of other things before you can call it a membrane, before it's fair to say it's a membrane. But a liposome can increase in size for quite a bit further than 2 to 20 nanometers. Now, the width of this is still like 5 to 9 nanometers. Like the actual width, the distance from this head to this head is somewhere like 5 to 9 nanometers. Let's talk about bilayers for a moment. What else is in bilayers? Proteins are in bilayers. We have other lipids in bilayers. If we recall, other lipids include cholesterol. We should also draw membranes and membrane proteins. So let's draw these things. We have our, oh good, the ink's back. We have our phospholipid bilayer here. Well, that's the start of it anyway. We've got some proteins. I have a protein like that, easy enough. I also have more phospholipid bilayer. I might also have a protein like that. I might also have a protein like that. I might also have a protein like that. There's all kinds of shapes and sizes of protein. After all, I've got 20 amino acids, and I can have many, many different lengths, and I have all kinds of different tertiary structures. So, membrane proteins will have a specific conformation of hydrophilic to phobic, and they have to remain in place in order to remain in place. So remember that this is non-polar. This is non-polar, and this is polar. And this is polar. And this is a polar solvent. This is a polar solvent. So that means that this region of the protein needs to be non-polar. And this region of the protein needs to be polar. The exterior of that protein. Same thing here. Polar, non-polar. Same thing here. Polar, non-polar. What about here? If this were a channel protein, where I am allowing, for example, water, which is a polar substance, through, or an ion like sodium through, which is a polar solvent, which is a polar ion because it's a charged particle, through, then this must be polar. This region right here must be non-polar. And this region must be polar. So there needs to be a specific conformation of polar and non-polar in order to stay anchored into these positions and in order to allow for the passage of stuff if it's a channel. Now, if you remember Unit 1, there was a term called consolidates. A consolidate is when a different type of macromolecule, when two different types of macromolecules combine together and they change properties. So, proteins that have a prosthetic group will be a consolidate. A protein with a prosthetic group is a consolidate. But it's possible with any given macromolecule as well. So glycolipids and glycoproteins, you have a lipid down here with a glycolipid, that's your glyco, that's your carbohydrate right there, attached to a lipid. You can have a glyceroglycolipid, where you have a carbohydrate with two fatty acid tails. You could have a sphingoglycerolipid, where you have some chain right there, and a fatty acid right there, and this is a carbohydrate right there. You can also have a glycoprotein. So this is a carbohydrate right here, and this is a protein right there. So whatever is the second word in a consolidate, that's the larger part of the molecule. So a glycoprotein, the protein is larger than the glyco part. A glycolipid, the lipid is larger than the glyco part. That's just part of your naming scheme. Fair enough? So let's draw one of these glycoproteins right here. Ta-da! Just one thing to point out, the point of attachment for these glycoproteins, which by the way are very similar to the glycogen aspect of it, is it's actually very similar to glycogen, but they often have a unique structure, and they are often used as tags for the major histocompatibility complex, the MHC. They can also be used as mucin, for example, which is an environmental control molecule. They increase the viscosity of the environment surrounding the cell by retaining water and the like. Now, you could also have a protein around here, by the way, which can serve a number of functions. Now, oh, by the way, I'm just going to draw a cholesterol molecule over there, OH. Another cholesterol molecule over here, just because I feel like it. All right. So, here's my final, putting it all together. That brings us to the idea of the fluid mosaic ring. And by the way, when we want to draw a glycolipid, excuse me, I'm going to draw it like this. I'm going to draw a regular old phospholipid with a glycogen molecule sticking out of it. So here's the kind of breakdown. Here's what you should be looking for in these images. You should be looking for and labeling certain things. You want to always label your hydrophilic heads and your hydrophobic tails and the phospholipid bilayer as a whole. So the phospholipid bilayer as a whole needs to be labeled. You want to label that. You want to label the phospholipid heads of the extracellular membrane surface or in the intracellular membrane surface. So this is the intracellular space. This is the extracellular space. You can always tell the outside of the cell from the inside of the cell by the presence of these glycoproteins and glycolipids. The sugar part of a glycoprotein or glycolipid is always going to be on the outside of a cell membrane. Always, 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 the sugar part is going to be on the outside of that membrane in your cartoon drawings. Integral proteins are fully integrated throughout the image. So they're all the way through. Integral proteins are fully integrated. Your peripheral proteins are in the periphery. They're only in one layer. Channel proteins will have a channel through them that can channel substances across the membrane. Now, glycoproteins are going to be globular proteins with a glycogen molecule sticking out of the top. You will also find cholesterol molecules as pegs periodically. Now, these can be drawn roughly, as shown here, and by me, because I'm not a good artist, but they can also be drawn as actual tetracyclic ring structures, like a good chemist. And did I miss anything? Nope. All right. Well, here we're also seeing the cytoskeletal elements. These are the filaments of our cytoskeleton. So that's not a problem. Oh, this alpha helix thing, that's just another protein. Don't worry too much about that. Oh, surface protein. A surface protein on the inside here. That's nice. That's not embedded in the membrane at all, and I did draw that. That's not embedded in the membrane at all, but that is just close up on the surface. I think I covered everything. Yep, we're good. So, here's what I want my cell membrane to be able to do. I want my cell membrane to be able to protect my cell. I want it to be able to identify my cell as belonging to the organism. I want my cell membrane to regulate fluid. I want it to regulate electrolytes. I want it to perform exo, endo, phagio, and pinocytosis. I want it to recognize signals. I want it to signal other cells. I want it to receive signals from other cells. I want it to be fluid. I want it to immobilize enzymes. And that's most of what I want it to do. So, as far as antigen recognition is concerned, this is part of recognition and cellular recognition and cellular signaling as far as identification is concerned. So you'll have your glycoproteins, and you'll have your glycolipids. You'll have your carbohydrate chains, which are glycogen chains, which are alpha-1,4 glycosidic bonded chains of carbohydrates, glucose, alpha glucose, with alpha-1,6 branches. These branches are in specific branching patterns of specific sizes and shapes. These specific sizes and shapes are going to be recognizable. They're going to be unique to that cell or to that organism, for example. So that's the major histocompatibility complex at work. The major compatibility of tissues. Histology is the study of tissues. So these act as antigens. Antigens are recognizable molecular structures recognized by cells. Recognized by cells in the immune systems. So what sorts of materials do I want to be entering and leaving my cells? I want water to enter or leave my cell. I want gases, ions, macromolecules, hormones, and neurotransmitters to be entering and leaving my cell. That's what I want to enter and leave. So how is this going to happen? Let's talk about hormones and neurotransmitters for a moment. Water, gases, ions, and macromolecules I can deal with later. But hormones and neurotransmitters, I want to talk about cell signaling for a second. Our signals are going to be ligands or environmental stimuli. So ligands are primary messengers, like hormones and neurotransmitters. They might also be environmental stimuli, like stretch or temperature or concentration inside or outside the cell. My receptors are going to be cell surface membrane receptors. These are going to be proteins on the cell surface. They could also be cytoplasmic receptors, which are proteins inside the cell's cytoplasm, big surprise, or nuclear receptors, which, guess where they are? That's right, in the nucleus of the cell. So, second messengers are basically everything in between the initial stimulus and the effect of that primary stimulus. So, here is, in a nutshell, cell signaling of a membrane-bound receptor. I have a hormone or an environmental stimulus. The receptor receives the signal. This is signal reception. Reactions produce second messengers. This is signal transduction. Then I have a response to this. An activation of cellular responses occurs. This is my response phase. Reception, signal transduction, response. Now, for ligands that are capable of diffusing freely across a membrane, a receptor-bound membrane simply will not do. You need to have receptors in your cytoplasm or in your nucleus, depending on where you need your hormone or neurotransmitter to go. If the hormone is a lipophilic messenger, lipophilic or nonpolar, it can cross the membrane by diffusion freely. Once it diffuses across, it will meet up with that receptor. That receptor will bind to and will then undergo the same system. it will receive the signal. It will perform signal transduction, either by using second messengers or directly interacting with the DNA itself. And then we will have a response where the DNA will change its transcription of RNA to a new sequence of events, and we will start forming new